Welcome to my channel of Biotechnology Tutorial and Research Lab. Title of my lecture today: Cancer cell inhibition by olive leaf and fresh fruit extract as anti-cancer biomedicine, or phenolic content and antioxidant properties in olive fruits and leaves. It belongs to the courses of nutritional or biomedical biotechnology. Background of this lecture: cancer cell is an important issue nowadays for cancer treatment. Scientists are trying to make treatment of cancer cell by using different treatment. Olive fruit extract benefits related alternative medicine, supplements and vitamins. Still now there is no proper medicine or not invented any medicine except therapy. That is why scientists are trying to produce biomedicine to control cancer cell damage. Many researchers are doing work on it. Olives, fresh olives and olive oils are good biomedicine for many diseases. Especially cancer cell can be inhibited by fresh olives. According to the scientist reports, due to the existing more antioxidant, it is meant oleuropein. This antioxidant prevent to make cell DNA damage and cancer cell proliferations. That will be effective, and cell damage can be controlled by using fresh olives. Many literatures found by using olive oil, but no literatures found by using fresh olives, except few, means one or two literatures. That is why it is most important for controlling cancer cell damage. We can see here the olive leaves and olive fresh fruits. Materials and methods we can discuss here. Olive fruit extract, according to the Juan et al. 2006, Spanish scientist, he invented olive fruits extract and used it to control cancer cell damage, and successfully he gained. So also other scientists, they used the same procedure and they also got success. They use chloroform and methanol extract and the fresh olive extract completely in vacuum in a rotary evaporator at 35 degrees centigrade. The extract from the skin of the fruits are analyzed by gas chromatography in different conditions. Then after that, they did the cell culture, ST29 cells. This is the one cells were provided by American types culture collections. Commercially, we can collect this type of cell now in the market. Then this is the human cells, cancer cell, and these cells cultured in the laboratory and use the extract and EDT and different biochemical process Olive fruit extract was dissolved in dimethyl sulfoxide. All cells received dimethyl sulfoxide to a final concentration of 2%. Then cell proliferations. St29 cells are seeded at a density of 5.10 to the power 3 cells per oil. So this is the standard. And media, they prepared the media for culturing the cell with the olive extract. Cell numbers were measured using the fluorescence multi-well plate reader. DNA fragmentations, they did the DNA fragmentations as a late marker of the apoptosis. It means the death of DNA cell, normal death, were determined by the stain in DNA, and they counted the cell percent. Result and discussion. It is believed to beneficially affected numerous biological processes. There is no studies on the effect of the minor components of olive fruits. So they demonstrated and extracted of the waxy coated of olive fruits, and they used it to culture the ST24 human colon cancer cell. From the result we can see here 25 to 75 micromole per liter. They got growth inhibitions. They used the chemicals of mesclinic and olenolic acid, and also they used the olive fruit extract, fresh olive fruit extract. And they mentioned that high concentration of olive fruits extract with full inhibition of cell growth, full inhibitions. So this is their findings and good results. So we can see here also other scientists, they explained the same result. Experiment 2, according to the 2014 olive juice extraction methods. You can see here olive leaves, leaf blendings. We did this experiment extractions method. First day extraction, it became green and then turned to the brown color of the extract. And then from olive fruits also we collected grindings first and then extracted juice, and extracted juice we preserved. This is in the laboratory we did this work, olive juice extraction method from leaf and fruits. So there is some references of scientists, those who have worked in this field. You can see here experiment 3, we can see fresh olive extract uses for cancer treatment by the inhibition of cell proliferations, biochemical, genomic, DNA and the antioxidant properties. They use the olive leaves, fruits and olive oil can be used for biomedicine for health benefits as well as anti-disease treatment. Nowadays olive leaf and olive fruit extract are being used for cancer treatment, showing different cell proliferation in the cell culture technology. Olive leaf extract significantly inhibited proliferations, and they use here the B16 mouse. They inserted the cancer cell into the mouse and then they fed the mouse of olive extract and see the result. Olive leaf extract treated B16 cells. This is mouse cell B16 cells. Are blocked in the phases G0 and 1 phases. This is the of the cell cycle. This is cell level. It was reported that the intake of olive oil has been shown to induce significant levels of the apoptosis, the death of cells which occur as a normal and controlled part of an organism's growth or development. Another scientist, Han et al., he did this experiment and he reported that anti-cancer properties have been shown in olive oil due to the phenolic compounds present in the olives. They use the phenolic compound present in the olives. Beneficial health effects of olive have been attributed for the presence of oleuropein and hydroxytyrosol. This oleuropein the most important. This is the component of antioxidants. That's why the fresh olive is most important to control the cancer cell inhibitions. Major phenolic compounds of olive oil are observed for its effect on growth in MCF7 human breast cancer. Also other scientists he used human breast cancer and he got the result that less cancer cell proliferations and then cell apoptosis, cell death and cell cycle he observed, and they also found the oleuropein decreased cell viability. Cell viability decreased, inhibited cell proliferations and induced cell apoptosis. Oleuropein means hydroxytyrosols decrease of the number of MCFL cells. This is the breast cancer cells. By inhibiting the rates of the cells proliferations and the inducing cell apoptosis. Han et al. 2009. So that's why the phenolic compounds of olive oil and leaves are a complex mixture of the components that includes 3,4-dihydroxy phenyl ethanol and also other components they found. So most of the scientists got the result from olive oil. Nowadays the researcher is trying to get the result from fresh olive fruits extract. So and olive leaf extract, both are important nowadays. In vivo and in vitro studies suggest that these bioactive components exhibits powerful antioxidant activity. This is the main components. It is present in free form and as a constituent of the complex molecules such as oleuropein in leaves and fruits. So some in vitro studies on olive leaves have shown that its extract can decrease blood pressure and dilute the coronary arteries surrounding the hearts. So this is also another scientist invented this. Moreover, hydroxytyrosol means it is also antioxidant, has been shown to have anti-cancer effect on human colon cell, means ST29 cells, and human leukemia HL60 cells. Other scientists found these reports. So we can see here many scientists they now started different research. Hussain et al. 2015 we studied the biochemical genomic DNA characterization nutritional content in fresh olives at different water level and found significant effect the biochemical nutrient content and DNA characterization at different water stress. Juan he also et al. 2006 to extract waxy coating of olive fruits, and he found uh, the positive results. The and anti-proliferative effect on ST29 human colon cancer cells. Materials method, the same material method we used. The data collection, we collected data. Fruits or grounds, mortar and pestles, and finally extracted juice. Juice were collected and kept in the freezer for short times. This is the dry olives and fresh olives. Two types of olive we used, and we extracted the juice from dry and olive juice. And also we determined different types of the nutrient contents: potassium, calcium, nitrate, sodium. DNA isolation, we did the DNA isolations, and we DNA quantifications and characterizations by agar-gel electrophoresis. So this is the method. In short, we can see here the method. Then analysis: antioxidant, polyphenol, biochemical, glucose, and inverted sugar content and determined. Finally, DNA isolation, quantification, sequence were done. Then the results of glucose, inverted sugar, nutrient content have been measured. Antioxidants also we measured. This is called the fall-in colorimetric method, and we found very good result. Cancer cell culture treated with extracted fresh olives. In this step we did not do, we just analyzed the nutrient content, DNA quantifications, and antioxidant. Result of discussion we have seen here, we got the positive result, and we got the DNA yield, we got higher in fresh fruit olives than dried olives. Then we got the invited sugar, glucose, pH, and total polyphenol olefin higher in fresh olives than in dried olives. We have seen here DNA identification of DNA band standard, and then there is not so much difference, but you can see here DNA band is little bit difference from fresh olives and dried olives. So we can see nutrient content also: nitrate, potassium, calcium, sodium. There is it was higher in fresh olives than dried olives. Cancer cell culture treated with extracted fresh olives. After the investigation of the antioxidant, biochemical content, genomic DNA, and nutritional properties, we concluded that and recommended our results which might be related to the cancer cell proliferation in the cell culture techniques. In further studies, it will be explained, but from the other research that handles our antioxidant and biochemical properties are standard for cancer cell proliferations. Discussions: many scientists have discussed, you can see here, and they gave their opinion. So most of the scientists explained that fresh olive fruits and olive leaves extract are better than olive oil because it contains more antioxidant, olefin. This is almost the discussion we can see here, and we published this paper in the European journal, and also other scientists published in different American and European journal and cancer related journal. So in conclusion, you can say our result conclude the fresh olive contain more polyphenol biochemical contents, DNA band, and nutrient content than dry olives. Therefore, fresh olive would be more effective for anti-cancer treatment. These are the references. Those who are working on oncology, means cancer studies. So you can see their names, their references here. So this is the topic. So we can see here. Finally, we can conclude that fresh olive oil, fresh olive fruits, and fresh olive leaf extract contains more antioxidant, which is most important to inhibit cancer cell proliferation. Thank you very much for listening, watching, and subscribing to our channel.